So our first speaker is here virtually. She is Marie-Claude Vohl, a professor at the School of Nutrition at Université Laval in Quebec, and she's one of Canada's 5 key strategic research chairs. Her chair is called Genomics Applied to Health and Nutrition. And her group has studied genes related to cardiovascular disease risk factors and the way in which these genes interact with various nutrients. And she specifically has worked on genes that influence the human metabolic response to omega-3s. So she has nearly 300 presentations going back to 2008, and her presentation today will cover known gene polymorphisms that drive biological responses to omega-3s and explain how best practices in nutrigenomics can help health practitioners to be able to provide precision nutrition advice. So, Marie-Claude, welcome.
Thank you very much, Helen. It's a real pleasure to be with you today, although it's only virtual. So I will first share my PowerPoint. Okay, so you should see my PowerPoint. And before starting to present on the genetic risk prediction for the effects of EPA, DHA on mainly on the plasma triglyceride response, I would like to thank the organizing committee for giving me this opportunity to present on this very I would say very interesting topic for me. That's really my topic of interest. So it's a real pleasure to present today. So before starting, I want to show you the American Society for Nutrition research priorities. And you see on this slide that the research on the variability in response to diet and food is one of their top research priorities. It's for achieving personalized nutrition with dietary recommendations that are tailored to each person's needs. So it's really anchored into the field of precision nutrition. So precision nutrition is the science. It's an holistic approach to developing comprehensive and dynamic nutrition recommendations relevant to both the individual and the population health. So the primary goal of the precision nutrition is really to optimize the metabolic response in individuals or population subgroups through tailored dietary approach to promote health and prevent and treat diseases. So for doing that, you really need to work. It's really a framework of research and practice that will consider multiple and synergistic levels of influence, including the dietary habits, the genetic background, the health status, the microbiome, the metabolism, the food environment, the socioeconomic and psychosocial characteristics, and the environmental exposure. So we have illustrated the concept of precision nutrition into this precision nutrition plate, And really it's a schematic representation of the main factors or to consider when approaching precision nutrition. So you see again the same factors. So you have the dietary habits, the food behavior, the nutrigenomics, microbiota. So you need to be able to study the complex interactions between all these main factors for being able to do a very good precision nutrition. So on this slide, I just want to mention that the United States, they just recently invested into the field of precision nutrition. So the NIH just recently invested $170 million for the Nutrition for Precision Health program. So it's a program that will be developed between the big All of Us study. So they will recruit 10,000 people from diverse backgrounds in order to create the tools to provide the individuals with personalized personal dietary recommendation, because they fully recognize that just like medicine, nutrition is it not a one-size-fits-all? So they clearly understand now, and it's really well accepted now by the scientific community and by the medical and nutrition community, that it's very important to understand how diet affects an individual health, and it's very complex, and it involves how the microbiome, metabolism, nutrition, genetics, and the environment, they all interact together. And until now, these complex interactions are still poorly understood. This is why much more research is needed in the field, and that's a nice investment that has been done in the United States to be able to develop more on this very interesting very interesting topic of precision nutrition. So today, I will speak more about the omega-3 fatty acids. So just a few facts very quickly. For sure, there is an abundant literature, but still there are some controversies and mitigated results that are depending on the study endpoints. There is also a substantial heterogeneity in between the studies when regarding to the study design, such as dose, duration, inclusion and exclusion criteria. But still, several randomized clinical trials showing the consistent hypoglycemic effect of the omega-3 fatty acids and several meta-analyses are confirming the plasma triglyceride lowering effect. So we designed the fatty acid sensor study that we call FAST to study the variability of the plasma triglyceride response to an omega-3 fatty acid supplementation. The ultimate goal was really to demonstrate that the large-scale omics technologies can be used in human nutritional studies and for the development of precision nutrition applications. So we designed, as I mentioned, a very simple study. So we put the participants on a running for 2 weeks, which is only a stabilization period. And during that time, the participants had to follow the Canada's food guide recommendation. For the next 6 weeks, they continue to maintain their dietary habits stable. And they followed the Canada's food guide recommendation, but they were supplemented with 5 grams of fish oil containing 1.9 grams of EPA and 1.1 grams of DHA. So the subjects for this study, they were aged between 18 and 50 years. They had a body mass index between 25 and 40 kilograms per meter squared. They were non-smokers, not suffering from metabolic disorders, and had not taken omega-3 fatty acid supplements 6 months before the intervention. And during the study, we collected the biological material at the different study endpoints. So we collected the plasma for doing metabolomics, the DNA for the genetics and epigenetic studies, the red blood cells for the fatty acid analysis, and also we collected the peripheral blood mononuclear cells for being able to do the transcriptomic analysis. So today we'll speak more about the genetics part and a little bit about the metabolomics analysis that we have done. So this slide is only to show that, as expected, the supplementation with the fatty acid or fish oil capsule, with this supplementation, we achieve a decrease in the plasma triglyceride levels in the entire cohort. So, as expected, we have seen a decrease in the plasma triglyceride levels. But what is interesting for us is really on that slide. So you see the inter-individual variability in the plasma triglyceride response to such a small and simple intervention, so to an omega-3 fatty acid supplementation. So you see that it's highly heterogeneous and that you have nearly 30% of the subjects that will be non-responders or negative responders, meaning that they do not decrease their plasma triglyceride levels when they are supplemented with omega-3 fatty acids. And this heterogeneity and the fact that a certain percentage of the population are non-responders has also been observed in the FinGen study realized in the UK by Dr. Anne-Marie Miniane and Dr. Phil Calder. So they had a similar intervention. They had an 8-week supplementation with 1.8 grams of EPA and DHA per day. And they also observed that 31% of the subjects they were considered as non-responders to the omega-3 fatty acids when we are looking specifically on the plasma triglyceride levels. So in order to understand the heterogeneity or the inter-individual variability, so we have performed a genome-wide association study using the human Omni-5 big chip from the Illumina company. So it's nearly 4 million of markers that have been genotyped for all of our study participants. And on this slide, so you see the genetic variation that achieve a statistical significance. So there are those genetic variations or SNPs that are here above the blue bar. So with all these SNPs, we calculated a genetic risk score by summing the risk allele for each of the study participants, and then we add a genetic risk score for each of them, and we were able to see a difference between the non-responders and the responders. So if you are a non-responders, so you will carry more risk allele, or the more risk allele you carry, the more chance you will have to be a non-responder. And this genetic risk score was able to explain nearly 22% of the variance of the plasma triglyceride response. And this is true after the adjustment of for the effects of age, sex, and BMI. And we replicated these results into the FinGen study with the collaboration of Dr. Miniane and Dr. Calder, because replication is very often an issue. We cannot have a clear conclusion without a replication into an independent study. So this is why we collaborated with Dr. Calder and Miniane and replicated in the FinGen study. In that study, the genetic risk score explained only 2% of the variance in the plasma triglyceride response. This is also after the adjustment for the effects of age, sex, and BMI. So that was only the first step. When you do your GWAS, the next step is to be able to do the fine mapping. And for doing fine mapping, so because this is the pathway that we need to follow after the first step, which is the GWAS. So after the GWAS, we are now here. So fine mapping. This is why we have done fine mapping using dense genotyping, mainly by imputation. So for doing this imputation, we use the 10,000 Genome Project database as a reference set and the impute2 algorithm. And on this slide, you will now see that we have been able, with imputation, to greatly densify the number of markers into the region of interest. So this is the region of chromosome 3. So the black dots, they are the SNPs that we add with the Illumina base chip, and the red dots, they are those that have been imputed. So you see that we have increased the density of the markers into the region of interest. So we add a couple of regions of interest that those are above the blue bar on our GWAS graph. So after quality control and using a tag SNP approach, which is using a SNP that is representative of the other SNP into the neighborhood. So you choose only the main one that is representative of the other one in the neighborhood. So this one is called the tag SNP. We ended up with 505 SNPs of interest. And after refining by using the stepwise models, again adjusted for age, sex, and BMI, we ended up with a set of 31 SNP that was able to explain 50% of the variance of the plasma triglyceride response after the adjustment for age, sex, and BMI. So when we computed this genetic risk score with these 31 SNP, again, by summing the risk allele for each of the participants, we see that we have now a better separation between the non-responders and the responders. Then we add only with our first GWAS SNP. So we have a better separation now and a higher prediction of an higher explanation of the variance of the plasma triglyceride response. So we again replicated into the FinGen study with the collaboration of Dr. Calder and Miniane. And we significantly explain nearly 4% of the variance after the adjustment of age, sex, and BMI. So fine mapping was effective to refine our previous genetic risk score, as it is for any complex trait. So there is no difference with a nutrition phenotype traits than with any other complex traits that are studied by the geneticians. And carrying an increasing number of at-risk alleles of the 31 SNPs confer a higher risk of being a non-responders to the omega-3 fatty acid supplementation. And then we can conclude that the genetic profile is an important determinant of the plasma TG responsiveness to an omega-3 fatty acid supplementation. and then could be used into precision nutrition application to target the subjects most likely to gain clinical benefits from the omega-3 fatty acids. So we wanted to see if we were able to replicate the results into independent study and into population with different ethnic backgrounds. So we collaborated with researchers from Mexico and from the Nestle Center to Switzerland. And we had access to a Mexican population that have been supplemented with omega-3 fatty acids. So they were supplemented also for 6 weeks with 2.5 gram per day of APA and DHA. And we have computed our genetic risk score into their study participant, and then we use a sliding window approach into which we were excluding subjects more and more and more and more. So we, at the very end, we were keeping more like the extreme, so those with a very clear positive or negative response. And the more we were, I would say, cleaning our data set, and the more we were able, a greater percentage of the variance of the plasma triglyceride levels. And in this study, we wanted to see if the genetic risk score was able to predict the plasma triglyceride responsiveness to either EPA alone or DHA alone. So with the collaboration of Dr. Benoit Lamarche, also from Université Laval here in Quebec City, we tested the capacity of our genetic risk score to predict the plasma triglyceride responsiveness to APA, and it was significant for APA. So the genetic risk score predicted the triglyceride responsiveness to APA, and was a trend was observed for DHA. So when we have done this replication, we have a decreased predictive potential into the replication cohort, but still the genetic risk score was effective in most cohorts and across ethnicities. And the differences that we observe between the studies of cohort, they can be explained by the changes in the SNPs, because for some of the studies we had to replace SNPs that were initially in our GWAS or imputed depending on the technology we were using to genotype their study participants. So we sometimes had to replace a SNP for one in linkage disequilibrium near one of our SNP of interest. So this may have changed a little bit our prediction capacity. There were also some differences in allele frequencies between the cohorts and the ethnicities, and some difference between the studies regarding the dose and the duration of the supplementation that was a little bit different between the different studies that we used for the replication. And then we wanted to go with more multi-omic approach. So today I will show you also the results that we obtained with the metabolomics, mainly lipidomics, that we have done with Dr. Matthew Piclot from the USDA. So with Dr. Piclot, we have been able to quantify the triglyceride species by mass spectrometry. And then we made a discrimination of the triglyceride species differing between the phenotypes, so differing between the responders and the non-responders, using SPLSDA with the MetaboAnalyst package. So you see on this slide, so this lipidomic approach, we measure all the triglyceride species, as I mentioned, and we have done a principal component analysis to identify the main component. And then when we look at the difference between these two components, we were able to separate the positive responders. You see they are all in red, from the negative responders, so those not being able to decrease their plasma triglyceride levels following the supplementation, and the non-responders being those subjects more in the middle with a more mitigated response. So we were able to have a relatively clear separation between the phenotypes. And where are the differences with the differences between the three phenotypes, so the positive, the non-responders, and the negative responders. These differences were mainly in these triglycerides with the low short chain fatty acid. And we see that with the supplementation, the long chain fatty acid increase, but this increase was observed into all the three groups, so those positive responders, non-responders, or negative responders. And when we integrated our genetics and metabolomic features using these 10 triglyceride species that were different between the responders and the other two groups, when we combine, we had a balanced accuracy of nearly 0.25, which means that we had 75 accurate classification of the subjects in the positive responders group when it was a really positive one, and 70. So it was 79% for the positive responders and 71% for the negative or non-responders. So by combining genetics and metabolomics, we had an accuracy classification of about 75%, which was considered good. So genomic and lipidomic analysis were very consistent, meaning that we were still able to separate and clearly identify the positive from the non-positive responders from the non-responders. And that these different omics data, they can complement one another. So these multi-omics approach, they are very promising for efficiently predicting the metabolic response to a dietary intervention. And we can use the same approach to predict not only the favorable health response, but also we can predict the adverse metabolic effects of omega-3 fatty acids regarding, for example, insulin sensitivity. And this is what we have also done and published in 2019. But the question now, are the consumer interested in nutrigenomics? So we published the knowledge and interest of the French Canadians regarding the nutrigenomics. And this was based on the response of an online survey that included 37 questions. The survey was realized in 2015, and although we recruited 2,238 participants, we had we have a valid questionnaires only for 1,425 individuals, and they are those subjects that we analyzed. And from these subjects, we have observed that 91% of them, they are willing to follow personalized dietary advice based on the genetic makeup. And this is an interesting percentage. And we see in the literature, in the different survey realized in Canada and in Europe since 2008, an increasing interest of the consumers over time for these personalized dietary advice. And on this slide, we see the results of an European studies realized with 8,000 participants. They wanted to verify if they were willing to pay more for personalized nutrition across Europe. And indeed, 30% of the participants reporting being willing to pay more for personalized nutrition than for non-personalized nutrition advice. And this was particularly true for men with higher income, but that was varying based on the socio-demographic factors. The question now is if the consumers are interested, are the health professionals ready to apply results issued from nutrigenomics? So we also verified the perspective of the registered dietitians of the province of Quebec, so one of the French part of the Canada. And we see that 77% of the registered dietitians, they knew about nutrigenomics, but 75.9% of these registered dietitians working in clinical nutrition in public care settings consider that they do not have the knowledge necessary to incorporate nutrigenomics into their clinical practice. This percentage was about 63% for those working in the private practice sector, which are those that are more prone to use these nutrigenomic tests into this practice. So this is why we observe a difference in between the two type of practice. And I just want to mention that for these nutritionists, for these dietitians and these health professionals feeling more confident to incorporate nutrigenomics into their clinical practice, they will need clinical practice guidelines based on using the genetic variations. And this is what we have published specifically for omega-3 fatty acid just this Monday, so on February 14, in the Frontier in Nutrition. So that's one of the example of the paper that we would like to see more in the literature in the coming months and year for being able to adequately prepare the health professionals to incorporate nutrigenomics into their clinical practice. So we see that the individuals are highly interested by getting DNA-based dietary advice for disease prevention and for health-related issues. And the development of precision nutrition applications will be facilitated by the training of the registered dietitians and the health professionals, so to make sure that they can use research findings used from nutrigenomics into their clinical practice. And I will end by very quickly acknowledging the work of all of my graduate students and my postdoctoral fellow and my research assistant for their dedication to this FAS project. I also want to thank all the staff of the Clinical Investigation Unit, all the collaborators and the international collaborators that participated to this project, as well as the funding agencies. So thank you very much, and I will be very happy to answer to your questions.
Thank you very much, Marie-Claude.